Yes, and for the question, so how to know whether we have completed the structure of particular protein or enzyme or not?
Say again.
How can we know that we have completed the structure of a protein or not?
Completed structure of the protein or not?
Yeah.
So, to determine the protein structure, have to be done in vitro. Okay. So basically, you purify the protein, and then you add them together. Okay. So for example, I want to determine the SCF complex. Okay. I could only using the purified protein to determine the structure. So your question is, how do I know SCF maybe have other protein? Maybe it's there, and then we don't know yet, right? For protein structure, we cannot do that. Okay? We could only know we suspect SCF, they bind each other, and then we do the so-called co-crystallization to determine these three proteins together, what the structure look like. Okay? Sometimes you could only have a model feeding process. You could only determine one, and then determine another one, and then you try to using computer model to feeding whether these two proteins interact, suppose it this way or that way. Okay, so we could only using known protein for determining the protein structure. We cannot say I purify whole complex and I want to determine the whole structure. We cannot do that yet. Hopefully one day we will be able to do that. But at this moment, it is impossible. Okay,
thank you for your lecture. So far you have talking about the ubiquitination of the CKI.
Mm-hmm.
So what will happen if the proteasome is inhibited?
Okay.
Even though if the protein is ubiquitinate at lysine 48,
Okay.
to target by the 26S proteasome, if the proteasome is inhibited,
Okay.
in some way, so
Okay.
what path it will go?
Very good question. If proteasome is inhibited, you can imagine all the substrate pull through that process will not be degraded. Okay, and then there's an enzyme, there's a drug, particular design for this type of study and then for chemotherapeutic use. One drug called Velcade. It's actually a proteasome inhibitor. And at that time, we tried to improve the treatment strategy because some of the protein, like I-kappa B alpha, I-kappa B, which is an inhibitor of N-kappa B, is very important to control the N-kappa B activity. And then because I-kappa B usually got ubiquitinated and degraded, okay, so the scientists and the pharmaceutical company come out with a drug to block that process to preserve I-kappa B. So sort of prevent N-kappa B activity. So you hit them with a proteasome inhibitor, I-kappa B indeed got preserved. Okay, thereby all the N-kappa B pathway got shut down. Okay? So if you have an inhibit the proteasome, all the potential substrate of the proteasome will be accumulating. Okay, thanks. Question there?
My question is because I am very interested in P53 functions in cancer. So you show us a lot of protein like COP1 and PIRH2, and those proteins can degrade the P53, but when is it expressed in the cell? Are those proteins expressed in normal cells or only in cancer cells?
Good question. I think I'm very impressed with all the students here that asked nice question about the regulation. The PIH2 or the CUP1, although they are the newly identified E3 ligase for PBD3. Okay, let's say we will count several E3 ligase for PBD3. There's an MDM2, there's a CUP1, there's a PIH2. Okay, the MDM2, our normal cell has MDM. Okay. A cancer cell have a highly expressed MDM2, especially in sarcoma. Right? Sarcoma has a high expression of MDM2. MDM2 is always expressed, not very often in other type of cancer. Only in sarcoma, maybe in other type of cancer, but not in well-known cancer like breast cancer or lung cancer. As for PIH2 or CUP1, okay, CUP1 in our lab we do a lot of study. CUP1 indeed is overexpressed in many types of cancer. And therefore, CUP1's capability in degrading PBD3, that's well characterized as you can see there. And then we know that CUP1 overexpression, because of these overexpression in many other types of cancer, will cause the downregulation of PBD3 and compromise PBD3 cell. Tumor suppressive activity. Right? PIH2 is not well studied yet, but just like CUP1, it's also been shown to overexpress in certain type of cancer. Okay? So, in a way so that's the reason we say every type of cancer could be different. Okay? The lung cancer and breast cancer, the mechanism for them to become cancer, all different. In the breast cancer, we have HER2 overexpression, we have the subtype of triple negative breast cancer, we have ER positive, PR positive breast cancer. By lung cancer, we don't have this situation. We do have PBD3 mutation, we have a KRAS mutation, we have the EGFR mutation. Okay? So different type of cancer, they have a different mechanism to become tumorigenic. And so that's the reason the chemotherapy drug target different kind of cancer. They are different. And we have to use different way to do that. And that's the so-called personalized medicine these days. Because everybody's situation is different. Okay? The guy who gets the lung cancer, we have to using certain treatment strategy. A person who gets the renal cell carcinoma or get the pancreatic cancer, we will have to use a different way to do the treatment. Yeah. And that's also pose a very important challenge for many of us, including many of you, that your effort and energy put in this area, we are expecting you will be able to do some contribution in the future. Okay? Hopefully, all these lectures will help you to form that particular aspiration to be able to do cancer research in the future. Okay? All right. Last question. Okay. So we will take a break for lunch.
Yes, another question I just sparked my mind. As far as concerned, as far as I can see in your lecture, that just like Saline have controlled some step in the cell cycle, but there is some other protein to control Saline, and there is other proteins to control the protein control Saline. So that is over and over. I wonder what is the beginning of the dark trend. Can you explain to me?
you repeat? I mean, I follow some part of your question, but I couldn't quite completely follow. Speak slowly. Yeah.
Okay, I mean that as far as in your lecture, there one protein also always have another protein to control the activities of that protein. So what is the beginning protein that controls the very beginning process?
Okay. Yeah. You ask all the same questions we scientists always want to ask all the time. Yeah. True. I mean, you find A protein regular B protein. What if the A protein be regulated by other protein? Indeed, that's the case. It's always the case. A will not be the starting point. Okay? There's an upstream of A, maybe number 1, number 2, number 3 protein there to be the upend regulator of all these signals. And 2, so that means that we have so much to do. Okay, nobody can tell you, oh, we finished all the biological questions of all these studies. No, it's always a question there. There's still remaining question need to be done. Okay, so you say the original beginning protein, who are they? Right? We ask all the time, and that's exactly the spirit of being a scientist. You ask that all the time. You know A regular B. What if other protein regular A? What are they? Okay? And A regular B, in addition to B, are there other proteins being regulated by A? Definitely they are. And you could see we have the SCF complex, we have the FBW7 regulating not just only cyclin E, regulating MYC, regulating C-Jun, regulating Notch. Only 4? It's a question mark. Maybe not only 4. There may be 5, 6, 7. Just remain to be characterized. Okay? So what regulates FBW7? Yes, that's a good question. So many things. Right now, we only know PPD3 is the one. Okay? How about the others? Remain to be characterized. Okay? Keep that spirit. That's exactly in the scientist. All right? Good? Thank you, everybody, for your attention.